Hello everyone, welcome back to the session. So in today's class we are going to study about the short answer question in the chapter called the type study of Periplaneta americana. Okay, so this Periplaneta americana is nothing but this is the scientific name of the cockroach. Okay, so we are going to deal with the short answer question in the with respect to this cockroach chapter. Okay, so the first important question will be write a neat labeled diagram of the mouth parts of the cockroach. So you should know that cockroach is coming under the phylum Arthropoda. So whenever we are studying about this phylum Arthropoda, the first important thing will be the word arthro means jointed. Okay, so the word arthro means jointed, poda means appendages. Okay, so we can call them as the jointed appendages. Okay, those animals which are having this jointed appendages we can place under the phylum Arthropoda. And another important thing will be another important characteristic features of the animals which is placed under the phylum Arthropoda will be the mouth parts. Okay, because they are having a different types of mouth parts. Those animals which are placed under the phylum Arthropoda, they are having different types of mouth parts. Okay, so that is why it is very important to study about the mouth parts of the cockroach. Okay, so this is what a diagram of this mouth cockroach. So just listen to this. See here, there is a labrum. Okay, so this is a labrum and this is an lacina, and the associated point will be this mandible. Okay, so this structure is mandible, and this is a maxillary pulp for chewing. Okay, so the most important characteristic features will be this hypopharynx. This looks like a tongue. Okay, the tongue like structure in the mouth part of the cockroach is called as the hypopharynx, and this structure is called as a labrum. We can call them as an upper lip. The upper lip is technically called as a labrum. Okay, so the mandible and maxillae, it is meant for the chewing purposes, and there is a paraglossa, and this is a labial pulp, and this is a glossa, and this is a gallia, and this is a lacinia, and this is the stipes and submentum. So these are all then permentum, prementum, and this is a submentum. And as it is very difficult to draw this picture, if you come across this question in the question paper, you just leave this question. Okay, just choose the other question and keep this question as a last option. Okay, because there is an important structure. If you are finding easy to draw this picture, it is your choice. Okay, but most importantly, only if you are sure with this diagram you can proceed, or not, or else just leave it. Okay, next describe the physiology of the digestion in the cockroach. Okay, so physiology of the digestion in the cockroach. So there is with respect to the human beings, in the digestion process you should know there is a mouth. Okay, mouth, pharynx, esophagus, stomach, small intestine, large intestine, rectum and anus. Okay, so this is what the digestive tract with respect to human. But in case of cockroach, which is placed into the phylum Arthropoda, you should know that is the initial passage of food. The initial passage of food through the pharynx and the esophagus, it is going to reach the crop. Okay, so where the food is storing. Okay, so after the stomach in the human, after the esophagus, the food has to reach to the stomach in case of human, but in the cockroach the food has been reaching to the crop. Okay, so digestion in crop, here in the crop the food has been digested. In the crop the food is mixed with the digestive juices. Okay, regurgitated via gizzard and the grooves. Most digestion occurs in the crop. So in human beings the most digestion will be occurring in the small intestine, but in case of cockroach the most of the digestion will be takes place in the crop. Okay, and filtration and transfer. Now partly digested food is filtered by the gizzard bristles. Okay, so we have studied a structure called gizzard. So the what is the point of having this gizzard means the gizzard having some bristle like structure. Okay, so that gizzard bristles, they are going to filter the digested food. Okay, they are going to filter the partly digested food. They are filtering Now they are passing through the stomodial valve into the ventriculus. Okay, then they are passed through the stomodial valve after filtering by the, after the food has been filtered by the gizzard bristles, it has been entered into the stomodial valve and into the ventriculus. Okay, now how the enzymes are playing a crucial role in the digestion processes in the digestion of the cockroach. Okay, so the first enzyme, important enzyme, will be the amylase. Okay, so this amylase is in case of human beings, this amylase we can call them as a salivary amylase in the mouth. Okay, so in the mouth the digested food has been the food has been digested with the help of an enzyme called salivary amylase. Like in the same way in the cockroach also they are having the amylase. So this is the involving in the conversion of, they are involving in digestion the carbohydrates. They are involving in digesting the carbohydrates. Okay, so the starch is nothing but a polysaccharide, that is nothing but a carbohydrates. So what is meant by digestion means the breaking down of complex molecules into the simpler molecules, that is called as a digestion. Okay, so here this amylase is converting the polysaccharide starch into the disaccharides. Okay, so initially polysaccharide will be converting into disaccharide. Further, the digestion is not stopping up to this conversion of this disaccharides. After that, the disaccharides is going to convert into the monosaccharides. Only if the polysaccharides converted into the monosaccharides, the digestion has been complete. Now, who is responsible for converting this disaccharides into monosaccharides? And here it is an enzyme called as an invertase. This invertase is also called as the sucrase. This enzyme is involving in converting the sucrose which is a disaccharide, then it is converting into glucose and fructose which is a monosaccharide. Okay, now maltase is an enzyme which is converting the maltose into the glucose. Okay, here maltose is a disaccharide which is converting into a glucose which is monosaccharides. Now lipases are involving in converting the lipids, which means the lipases are involving in digesting the lipids. So see, in case of carbohydrates, the polysaccharide has to be converted into the monosaccharides. Okay, in case of proteins, the protein has to be broke down into the amino acids. In case of lipids, that has to be broken down into the fatty acids plus glycerol. This is what a digestion is. The complex molecules has to be converted into the simpler molecules. Okay, so in case of carbohydrates, this amylase, invertase, maltase comes into picture. In case of this proteins are getting digested by the proteases enzyme. Okay, proteins are getting digested into the digested by the enzyme called as the proteases. Okay, so that the proteins may be converted into amino acids. Now the lipases they are converting the lipids into fatty acids and glycerol. Okay, cellulase are the enzyme, they are secreted by the hindgut microorganisms. See, you should know that this cellulase is absent in case of human beings, but in the cockroach it is present. Okay, the cellulase, thus they are secreted by the hindgut microorganism, digest the cellulose into the glucose. So this is very important, and this enzyme is absent in case of human beings. Now, after the food has been digested by the enzymes, it has to be absorbed. Okay, so now that is what an absorption. Now all the digested food has been absorbed. The undigested food that has to be eliminated with that process is called as an egestion. See, digestion means the breaking down of this complex molecules into the simpler compounds, that is called as a digestion. Absorption in the sense the digested food has to be absorbed by all the cells of the body. So that is why the complex molecules has been converted into simpler molecules, so that all the cells has been very easy to absorb all the simpler molecules. Okay, so that is why digestion is taking place. Because it has to make easier way for the cells to absorb all the nutrients. Now all the undigested food has to be eliminated from the physiological systems, and that process is called as an egestion. Okay, so in the ventriculus, the digested food is absorbed. So this structure is very important with respect to this cockroach absorption. Okay, so the undigested food moves into ileum. Ileum, which is nothing but an intestine, colon, and then rectum. Okay, these are all the parts of an intestine. Now water has to be reabsorbed by the rectal papillae. Okay, so here water is necessary for our body to eliminate the waste. Not only to eliminate the waste, for most of the physiological function, water is mandatory for the whatever be the organisms be. Okay, and also for the elimination of waste, water is necessary. And all the organisms, most of the organisms, those who are uricotelic and ureotelic animals, they are cautious in eliminating the water. Why? Because water is very essential for other physiological functions, not only eliminating the waste. Okay, so what are all the waste to be eliminated? So the water is necessary, water is sufficient just to eliminate the waste. Okay, so water has to be reabsorbed, the water has not to be eliminated more. Okay, so all the physiological, all the organisms are has to be cautious to eliminate most of the water. So just to eliminate the waste, the water is sufficient. The water has to be get out from the system just to eliminate the waste, not more than the waste. Okay, so that is why the water reabsorbed by the rectal papillae. Okay, now remaining material is defecated as dry pellets through the anus. So through the anus, all the they are defecated as a dry pellets. Okay, so this is called as an egestion process. Okay, so this is what a physiology of digestion in case of cockroach. Now coming to the next question, that is draw a neat labeled diagram of the salivary apparatus of the cockroach. Okay, so the salivary apparatus of the cockroach, you can easily draw this picture. Once if you practice 2 to 3 times, it will be easy for you. See, just listen to me. There is an hypopharynx. So as I have mentioned you before, the tongue line structure is called as an hypopharynx. Okay, so this is an efferent salivary duct. Okay, so this is an efferent salivary duct where the food has been passed through. Okay, after that, this is a median salivary duct. The structure is called as a median salivary duct. And above the median salivary duct, below the efferent salivary duct, that structure is called as a common receptacular duct. Okay, and after that, here there will be a receptacular duct, and here there will be a common salivary duct. So this is a common salivary duct that divides into 2 lobes. Okay, and this is called as the lobes of the salivary gland, and here also we can see the lobes of the salivary gland. So this is what the salivary apparatus. So in the salivary apparatus of the cockroach, we can see the hypopharynx, and the receptacular duct is very important, and the receptacle is very important, and the salivary duct and the lobes of the salivary gland. So these are all the parts of the salivary apparatus in case of cockroach. Now, one of the most important characteristic features of arthropods is ommatidium. Okay, so ommatidium, in the sense, it is called as a compound eye. So in the eye, we can classify them into 2 types. Okay, simple eye and compound eye. So this compound eye is also called as the ommatidium, and this is the structure which is present only in the phylum arthropods. Okay, so in the compound eyes, which is also called as an ommatidium, what are all the structure which is present in the ommatidium? Okay, so this ommatidium will be the structural and functional unit of the compound eye. Okay, so just listen to me. This is a cornea. The outer cell layer is called as a cornea, and below the cornea we can see the Carnegian cells. Okay, so below the cornea there is a structure like this, that is called as a Carnegian cells. Okay, and below the Carnegian cells there is a cone like structure, we can call them as a cone. Okay, inside the cone there is a crystalline cone. This structure is called as a crystalline cone. Okay, so in the crystalline cone, So this is a cone-like structure, and that cone, in this pigment is called as, so in the cone we can see the pigments, iris pigment sheath. Okay, so this is called as an iris pigment sheath, and this is called as an retinal pigment sheath. Okay, so pigment is very very important. That is why they can differentiate the, then only they can differentiate the colors. Okay, now after that there is an important structure called as a rhabdome. Okay, so this is what a rhabdome, and this structure is called as a retinular cells, and below structure this is called as a basement membrane. Okay, and below the basement membrane we are having the nerve fibers. Okay, so these are all very 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 important. Okay, so the structure of ommatidium, so in the ommatidium we are having this rhabdome, retinular cells, basement membrane. Below the basement membrane we are having the nerve fibers. Okay, so the first structure we are going to see one structure one by one. So the cornea is the outermost part. Okay, so this is an outermost part. This corresponds to the hexagonal facet of the compound eye. So the outermost part of the compound eye is called as a cornea, and this is the hexagonal facet. Okay, and this is biconvex. Okay, this is biconvex, and this has a transparent cuticle. As it is only transparent, the light can easily pass through. Okay, so if it is translucent or opaque, the light cannot pass through fully, or light cannot pass through it. Okay, so only if it is a transparent cuticle, the light can easily pass through that layer. Okay, and this cornea is secreted by the specialized epidermal cells. Okay, so this cornea functions as a refractive region of the ommatidium. Okay, so this is very important. Cornea is acting as the refractive region of the ommatidium. Okay, so this is a cornegian cells. This cornegian cells is also called as a lenticular cells. Okay, so here it is made up of two transparent epidermal layers. That is the one responsible for secreting the cornea. Okay, so we are having the cornegian cells, which is also called as a lenticular cells. So here these cells are made up of two transparent epidermal cells. So that cells are involving in secreting the cornea. Okay, so this cornea is functioning as a refractive region. Again, this cornea is a transparent cuticle. Okay, so later form the primary pigment sheath or the iris pigment sheath. Okay, so as I have mentioned you before, this cornegian cells form the primary pigment sheath. Okay, so this primary pigment sheath is also called as the iris pigment sheath. Okay, now vitreae. So this vitreae is also called as a cone cells or the Semper cells. Okay, so the cone cells is also called as a vitreae or the Semper cells. Again, this vitreae is made up of the four transparent conical cells located below the cornegian cells. So you should know that above layer is called as a cornea, below the cornea we can see the cornegian cells, and below the cornegian cells we are having the vitreae, which is called as a cone cells, and that cone cells is made up of four transparent layers. Okay, this surrounds and secrete the crystalline cone. Okay, so the crystalline cone was surrounded by four transparent conical cells, and that is called as a vitreae. Okay, so next, what is mean by this crystalline cone? So this crystalline cone, again it will be a transparent. Okay, so this crystalline cone was secreted by the vitreae. Okay, so this vitreae is the one secreting the crystalline cone, and this is surrounded by the dark primary pigment cells. Okay, so we have studied two pigment cells. One is primary, that is iris pigment sheath, and another one is retinal pigment sheath. Okay, and this crystalline cone was surrounded by a dark primary pigment cells. Along with the cornea forms the dioptrical focusing region. So this is another important thing, dioptrical region. Okay, focusing light onto the next part of the ommatidium. See here, ommatidium, this is a single, as I have mentioned you before, nephron is a structural and functional unit of kidney, and the kidney has millions and trillions of nephrons. Like in the same way, this ommatidium will be the structural and functional unit of compound eye. So like one ommatidium, we are having many ommatidium. Okay, now retinulae, so this will be the innermost elongated cells, 7 in number. How many retinulae is present in the ommatidium? 7 in number. Okay, so rest on the basement membrane. So the basement membrane gives rise to the retinulae. So now each retinulae have the microvilli forming the rhabdomeres with photoreceptor pigments. So only you should know that in the retinulae we are having the photoreceptor pigments. Now this rhabdomeres fuse to form the rhabdome. So this rhabdome is an important structure. This is the one involving in central light receiving structure. So the light is going to receive. So see here, the light is entered through the cornea and the light will be received by this retinulae. Why? Because this rhabdomeres, they are having the photoreceptor pigments. Many rhabdomeres together form the rhabdome. So this rhabdome is the one central light receiving structure. This retinulae, they are having the nerve cells. Another important thing, the nerves which is present in the eye is called as an optic nerve. Their fibers form the optic nerve to the photocerebrum. Together with the rhabdome, they are forming the retinal receptor region. Why we are saying this as a receptor? Because someone has to receive the signal. So that is why we are calling them as the receptor. Okay, and we are having the secondary pigment cells. Okay, so the secondary pigment cells were surrounded by the receptor region. So this is the one involving in the absorption of light. Isolate each ommatidium from one adjacent cells. See here, one ommatidium they are receiving a light, another ommatidium they are receiving the light, another ommatidium they are also receiving the light. So this secondary pigment cells, they are involving in isolating each ommatidium from the adjacent ones, because each ommatidium they are going to receive a light. Okay, so this isolation was done by the secondary pigment cells, then form the retinal pigment sheath, right? So that's all about the short answer question with respect to the cockroach. So in this lesson we have studied about this 4 short answer question. That is, first one is the structure. Okay, mouth parts of cockroach, and the second question will be the physiology of digestion, and the third one will be the salivary apparatus diagram, and the fourth one will be the structure of ommatidium. And this fourth question is very important. Okay, so we'll meet you in the next class. Thank you so much.